Hi, are you here for the metabolic engineering introduction session? Hmm. हाँ, வீடியோ ஆஃப் மியூட்ல இருக்கு. மூணு பேர் தான் வந்திருக்காங்க. அதான் சொன்னேன், இது ஒரு interaction session for discussing your queries. Please unmute and introduce yourself while others join அப்படின்னு சொன்னேன். எவனும் ஒன்றும் பேசல. ம்? அது வந்து இன்னும் 2 நிமிஷம் ஆகட்டும், 6:10 ஆகட்டும் வெயிட் பண்ணிட்டு இருக்கேன். எடுக்கிறீங்களே? ம், தான். ஃபேன் குறைக்கணுமா? ஆஃப் பண்ணிட்டா? ஷியரா? பெட்ஷீட் பேபியா? ம், சொல்லுங்க சொல்லுங்க. ஆஃப் பண்ணிட்டா? ஓகே. அவ்வளோதான் இது இருக்கு. சரி, நான் லைவ் போகிறேன். பார்ப்போம். தேங்க்யூ. பை பை. யா, கேன் யூ ஹியர் மீ? Hello. Can someone hear me? Ayush, Srishti, Tanisha.
Yes, you're audible.
Okay. Is my screen visible? Do you see a PowerPoint presentation?
No.
1 minute. Yeah, now you should be able to see my screen. Yeah, and my PowerPoint presentation is here. Yeah, because this is the first session, I thought we can just have an introduction, a short introduction. I'm Shankar. I am a PhD student from IISc Bangalore. I'll be the TA for this course, and we'll be meeting every Mondays from 6 to 8 to discuss your queries, and also we'll be trying to solve assignment problems so that will help you to solve your assignment problems in this for this run of the NPTEL course. We can start with a small introduction with whoever is available. You can go ahead. This session is supposed to be interactive, so it's better that a few of you talk. Okay, if Okay, we'll start with the course. If you have any questions with any of the lecture, you can maybe put that across through the chat box. We can wait for a few minutes. If not, then I'll go with a few sample questions that we can try solving. You will be given a few minutes to solve, think about each question, and you can discuss with me if you have any doubts. These questions are aimed to help you think about the problem so that your actual assignment gets easier. Okay, if there are no questions, we can start with the sample problems that we have for the assignment for week 1. So the first question is, what does not hold true for computer-aided synthesis of biochemical pathways, which is BNICE? And the options are, it considers the chemical structure of the substrates and products, it has a non-generalized algorithm, it does not consider the enzyme rules, or de novo design of enzymatic pathways. You can think about the answer and you can maybe type in the chat box. We'll wait for a few minutes and then we'll discuss the answer. Does anyone want to answer? You can type in the chat box or you can say it out aloud.
Computer aided
Sorry,
synthesis of I think
can you come again?
it has a non-generalized I think B is the correct answer for that.
Okay, and you can maybe take a look at it again if something else also does not hold true for it.
Mhm. Enzyme rules. I'm not getting what do you mean by enzyme rules means.
Okay, so be nice. It's a database. You can type benice.ch and check. That's the URL. So what this takes into account is for any given pathway, what is the substrate, what is the product, what is the enzyme, and how this enzyme acts on the substrate to give the product. So you can call that as the enzyme rule as well. So if you go to lecture 5 at this particular time point,
Mm-hmm.
around 14 minutes, they talk about uh, Be Nice, which is the server, and they say that all of these things, which is a generalized algorithm considering the chemical
And set of
structure,
enzyme rules.
yeah. So you have different kinds of enzymes, hydrolases, lyases. There are 8 classifications of enzymes according to enzyme classification system, and
Mm-hmm.
not one enzyme can act on all substrates. So those are considered the enzymatic rules. And incorporating that into the answer, the right answer here is E, which is BNC. It considers, it has a generalized algorithm, it does consider enzyme rules. Was that clear? Was that helpful?
Yeah, clear, clear.
Okay, yeah. Others, if you have any doubts, you can start talking like Surinder did. Uh, just to make you guys aware, this also has an attendance. So at the end of the lecture, I'll be taking an attendance of who all has attended. Yeah, yeah. The next question is, um, what are seed metabolites? Seed metabolites refers to externally acquired molecules. Option A. Internally synthesized molecules. Option B. The first metabolite of the rate limiting step. Option C. None of the above. Option D. You can think about it and either answer or write your answer in the text box. Ayush, would you want to go answer for this question?
A.
Externally acquired metabolic molecules. Okay. Others? Do you?
Raw material, no? It should be raw material.
Um.
And first,
So yeah, Ayush. 1 minute, Surinder. I would allow him to answer. Yeah, Ayush.
but I also think it's A. So
It's A.
because
Huh. Because?
because seed metabolites generally refers to a externally acquired synthesis.
Hmm.
Um, so
Yeah. Yeah, like both of you told, like Ayush and Surinder told, seed metabolites are those metabolites that cannot be naturally synthesized within a cell. So unless that is provided in the media to cells, the pathway cannot take place. So these are the starting points for synthesis of other larger molecules or conversions, bioconversions. So the answer is option A. Uh, the next question is, what is heterologous expression of gene? Uh,
Yeah, it's a B.
huh. uh, we'll also ask others if they what they think about. Is Tanisha there? Or is Shrishti there?
Yes.
Yeah. So would you want to answer, both of you?
Uh, is
What
it
is
option B?
heterologous? Option B. Okay. And Tanisha?
Sir, option B.
Okay. Why do you guys think it's option B? Would you want to like justify any of you? Yeah, you can go ahead, Tanisha. Yeah. Hi, Swati. Uh, can you introduce yourself?
Hello.
Yeah, you're audible.
Yeah. So I am Swati Lakshmi Bzilal, currently studying BTech Biotechnology and Biochemical Engineering, third year in Mohandas College of Engineering and Technology, Trivandrum, Kerala.
Okay. Okay. I am Shankar. I am a PhD student from IISc Bangalore. I'll be the TA for this course. So in this particular session, we'll be discussing some few sample problems that are related to week one of your lectures. So
Okay.
we started going through a couple of questions. This is the third question. The question is, what is heterologous expression of gene? The options are expression of a gene or part of gene in host organisms that naturally have the gene or gene fragment. Expression of gene or part of gene in host organisms that naturally do not have the gene or gene fragment. Option C, both A and B based on environmental condition. And option D, none of the above. Would you want to answer for this question, Swati?
I think option B is the answer.
Huh, okay, okay. And what about others? Ayush, you, mm,
Yes, sir.
oh,
It's B.
okay, yeah, like all of you told, heterologous expression means expression of a gene that was not naturally present in an organism, but you take it from an organism and then express it in a different organism for higher production. We'll discuss the questions that we had previously discussed once we complete all the questions, Swati, because others had attended that.
Okay, okay.
Yeah. The next question is, which of the following are not involved in the production of microbial cell factories? And the options are bioinformatic and mathematical modeling, which includes qualitative analysis of the pathways involved in the product formation. Option B is identification of the specific genes and their modifications through different recombinant DNA technology and allowing to alter the metabolic pathways of interest. Option C is flux redirected approach towards product of interest. Option D is A and C. Option E is none of the above. So you guys can think for a few minutes and start answering after that. While you guys are thinking, it will be helpful if uh, you guys can introduce yourselves.
Hello.
Yeah, Surinder, you can first
Namaskar ji.
introduce yourself. You and then others can follow you while we think about this problem as well. Yeah, you can go ahead, Surinder.
Hello.
Yeah.
Good evening, all of you. Good evening, all of you.
Mm.
Uh, Actually, I'm working as a faculty in
Okay.
Sharda University.
Okay.
Okay, at Sharda University, Greater Noida in NCR, Delhi.
Okay. Mm.
Okay, and I have completed a PhD in animal biochemistry
Okay.
from National Dairy Research Institute, Karnal.
Okay. Mm.
Okay, and overall, I have the means approximately 6 years of teaching experience.
Okay.
I found this course as a faculty development program.
Mm.
Okay, AICTC approved.
Mm.
Okay, and it is somewhat knowledgeable to me also. That's why I joined this course.
Okay.
Thank you.
Thank you. Ayush?
Yes, sir, I'm Ayush Chaudhary. I'm a student of MIT University, third year biochemistry, BSc Biochemistry.
Mm. Okay. Tanisha?
Good evening, everyone. I'm Danisha Sharma. I'm a student of Amity University. I'm studying BSc Honors Biochemistry and I'm in third year.
Mm. And Shrishti?
Hello everyone, I'm Srishti Banerjee. I'm in my second year in biotech engineering in NIT Rourkela.
Mm. Okay. So now that we are done with the introduction, yeah, who would want to answer this question? Would anyone want to volunteer?
Sir, none of the above,
None of
because
the
sir,
above. Huh.
None
all
of
we
the
are.
above.
Hmm.
It's E.
Okay, I
Option
would
E. Option.
option E. Okay. Would someone want to explain why they think none of the above? You, I,
So because
because we have,
all
because 1 minute, Ayush, because we have limited number of people, I think each of you can explain why one particular point does not, uh, you think it's not true. Yeah, you can start with Ayush or Surinder. Yeah,
sir,
you,
because
anyone.
generally all three option are generally involved in the production of the microbial cell factories. So, sir, I think that is not possible. So none of the above.
Huh, would you want to talk about any specific point of how it
Uh,
is specifically used in microbial cell factories? You can choose whichever you want and then try to say why you think that particular element is needed in microbial cell factories. If you think,
uh, s- sir, I need to think about, sir.
okay,
I'm not particular.
okay. Surinder, would you want to try?
Yeah, cell factories means source of the chemical. So here microbes are considered as a sources of the chemicals,
Right,
metabolites.
right.
So to increase the production of the metabolites or chemicals
Hmm,
using the microbes, we
hmm,
have to alter their genes also. We have to increase the efficiency of the genes.
right.
Okay, so
Hmm,
that production can be enhanced. And recombinant DNA technology help in the enhancing the efficiency of the genes.
right,
Okay,
right.
and bioinformatics tool
We can we
helps.
can we can allow someone else to answer for this the other options. Uh, Others, Srishti, Tanisha, Swati, anyone would you want to go?
So the option C says flux redirected approach towards a product of interest.
Yeah.
So basically, metabolic network is very, very complex.
Hmm.
So if you take the carbon flow the carbon molecules will be like distributed throughout in different pathways, like the pentose phosphate pathway or glycolysis or pepcycle or whatever may it be. So if you want to produce a product of interest, you have to bring all those, you have to make sure that the carbon molecules will flow through a particular reaction so that that particular product is produced.
Right. Yeah, that was correct. And option A is remaining. Srishti or Tanisha, would any of you want to attempt that?
It's basically like using mathematic like data. So you compile all of the data and use perhaps like computer science techniques to figure out like model uh, a process or something using it. I'm not entirely sure.
Okay. Would anyone else want to try, or should, yeah, if anyone else can attempt? Otherwise, I would explain for option A. Okay, for option A, whatever Srishti had told is partially right, but apart from what she had told, here mathematical modeling would involve kinetics of the enzymatic reactions, which are also taken into account. And bioinformatic analysis would mean that comparing pathways across different organisms at the protein level or at the gene level. That is also important to understand why genes or proteins from specific organisms are having higher activity, producing higher product, which you mathematically model through kinetic equations. Yeah, so we'll go to the next question now. The question is, the molecular biology techniques involved in the process of metabolic engineering are generally used for option A, blocking competing pathways, option B, decreasing enzyme specificity, option C, downregulating the expression of the regulator, option D, none of the above. You can take a few minutes to think and then maybe switch on your mic and start answering why one or none of the above of the options are right or wrong. You can take turns.
I think it's A, blocking the competitive pathways.
Okay, why do you think so?
A, answer A.
Uh, yeah, why do you think so? You can give an explanation for the same.
Because we have to increase a particular product. Na? For
Right,
example, we want to increase the production of A.
right.
Okay, if from one metabolite two molecules are forming A and B,
Okay.
so we have to stop the synthesis of the so that production of the A can be enhanced.
Hmm. Okay. Ayush, what do you think? Swati? Sorry, my mic was switched off. Ayush, what do you think?
Sir, I also think A.
Okay. And Swati and Srishti, do you have any opposite thinking or ideas? Do you guys also agree with option A?
Yeah, agree.
First, when I read the question, I was like kind of confused.
Hmm.
Still,
Okay, you can share what you think. We'll try to resolve the confusion.
so the third option, down regulating the expression of the regulator. So
Huh.
the regulator molecule is associated with gene expression like the operon concept or something
Yeah.
related to.
So in you can consider it that way as well. That is one way of thinking about it.
So down regulating the expression of a regulator means the regulatory protein will not be produced.
Maybe, or will be produced at a lesser extent than usually, than it is at the normal cases.
Oh, okay, okay.
So,
Option B cannot be the answer, right? Because if you reduce enzyme specificity means the product will not be produced.
you okay? Okay. I'll clarify that. So, you can reduce an enzyme which has specificity towards a particular substrate, but if it is competing, for example, there are two enzymes that are competing for the same substrate, then to avoid competition from uh, because of one enzyme which is not of interest to you, you can reduce that enzyme's specificity. You can think about it
Okay.
that way as well. But the question here is how are molecular biology techniques involved in this? So, if you think about molecular biology techniques, you can think about techniques related to cloning. Cloning that would involve taking upregulating a pathway or downregulating a pathway. So, to decrease enzyme specificity, you need to know the structure of a particular enzyme and you need to mutate the particular enzyme. To block competing pathways, you need to remove the enzyme from the system itself. It is called as knockout. And downregulating the expression of a regulator. Here they don't say what a regulator is, whether it is an activator or a repressor. So that also can be done with molecular biology techniques. But with respect to metabolic engineering, where we are interested in increasing product formation, the appropriate answer here would be blocking competing pathways, like it was mentioned by Surinder and others. Um,
Oh, okay, okay. Thank you for clarifying, madam.
huh. Hi Kritika, I am Shankar. I will be the course TA and we'll be meeting every Mondays on 6 to 8. You, I am a PhD student from ISC Bangalore. You can introduce yourself.
Good evening, sir. This is Kritika Chauhan. I'm a BSc Honours Biochemistry student from Amity University, and this is my last semester.
Okay,
Thank you.
okay. So in this session, we'll be discussing a few sample problems which will help you think and in general is supposed to help you solve your actual assignment problems that would have been opened now on the SWIME website. So this will be an interactive session. So Kritika, you can also start answering with the questions. So we were discussing few sample questions. Uh, would you want to attempt this question that you see on screen? The molecular biology techniques involved in the process of metabolic engineering are generally used for option A, blocking competing pathways, option B, decreasing enzyme specificity, option C, downregulating the expression of the regulator, option D, none of the above. Would you want to try attempting this, Kritika?
Yes, sir. So, sir, according to me, both option A and B can be correct, but whatever I have studied till now in metabolic engineering, the answer should be one, because we block those pathways whose product which we don't require. Uh, so I think option one is correct, sir.
Yeah, yeah, that's right. Hmm. Yeah, so now we'll go to the next question. Uh, the question is understanding of the complex network of enzymatic reactions that compose cells metabolism requires knowledge of option A, enzymology of participating enzymes, option B, structure and behavior of the pathways that connect enzymes, option C, both A and B, option D, only A. You can take a few minutes to think about this question and then you can raise your hand. Then we'll try answering the question. We can wait for a couple of minutes and then we can answer this question. Kritika can go first after a couple of minutes.
I, it's a C, I think. C.
Uh, 1 minute, Surinder.
Okay,
I wanted
okay,
to
okay,
allow Kritika to answer this question.
okay, okay.
We'll listen to her answer and then we'll discuss your answer as well.
Sir, can I answer now?
Yeah,
Sir, according to me, it's option C because when we study about the network, so we consider enzymes as well, which are participating in that pathway. As we know that metabolism pathways are interconnected, so by manipulating them, we can enhance the productivity. And second is structure and behavior of the pathways that connects enzymes. So sometimes we can connect some groups to a structure and by studying their behavior also we can manipulate the pathway as well.
okay. Um so the question here is understanding the complex network. So to understand the network, do you think you need to know enzymology of enzymes? Sorry.
Yes, sir. I think it's important. Like during my first week, I have studied which acetyl coenzyme was
Mm,
forming different products with the help of different enzymes.
mm,
So if we will not consider that, so that means somehow we will be lacking in that knowledge and we won't be able to get the specific product which we are required.
right. Good. Surinder, would you want to share your opinion?
Yeah,
Yeah. So you said it's option
C.
C. So you also
Uh,
think enzymology is important to understand the network, is it?
yeah, yeah, yes, sir. Because the complex network, na,
Yeah.
metabolism is integrated. So we
Hmm.
have to study the enzyme kinetics also. We have to study their Vmax Km value also.
Hmm. Hmm.
Okay,
Hmm.
that's why I think it is the C.
Okay. So the suggested answer here is
Because some enzyme having the
yeah. You can say because some enzymes have.
uh, because enzymes having the different affinities for the different substrates. Na? So we have to consider this factor also.
Yeah, but the
For example, if we
question.
consider ethanol and methanol,
Hmm. Yeah, to consider
so
ethanol and
enzyme
methanol.
alcohol dehydrogenase having the more affinity for alcohol dehydrogenase enzyme having the more affinity for methanol. So we
Okay.
metabolize the methanol first.
Right.
So that's why I consider enzymology. We have to study enzymology also.
Okay, that is to in general about pathways, but here they say about the network of the reactions. Like whatever you guys say is right. Both A and B are needed to understand. Huh. The next question is random
So, sir,
mutation.
the previous
Yeah,
answer for the previous question will be option C, right?
yeah, yeah.
Or
It is option C. Yeah,
okay, okay.
yeah,
Uh,
yeah. Sorry, who was that?
I have one query.
Yeah, sure.
What was the answer of last to last question? Blocking.
This this question.
Is this one
Yeah,
only answer?
blocking competing pathways.
Okay, thank you,
Because
thank you.
the theme of metabolic engineering itself is to enhance pathways. So directly we focus on using molecular biology techniques that will help linearize the pathway rather than diverge it and split it into many different sub pathways. So that way it's option A. And yeah, we are done with this question. The next question is random mutagenesis is the focal theme of metabolic engineering. There are two options. We'll wait for a couple of minutes, think about it, and then start answering. Yeah. Uh, we'll wait for a couple of minutes and then see if anyone else wants to answer. Yeah, this time instead of getting the answer, let's just go through the question. Kritika, do you know what random mutagenesis is? Is Kritika there? Okay, we'll go with Swati. Do you know what random mutagenesis is?
Sir, I think random mutagenesis is a phenomena by which we bring about mutation to the genome, like random mutation. You are not concentrating a particular gene. Okay, you are not
Okay.
concentrating to bring mutation to a particular gene. You are just exposing that particular microorganism to certain mutagens so
Hmm.
that mutation can occur. It can occur to the desired gene or some other random gene.
Hmm.
So I think that is random mutagenesis, and I think that
I don't want the
answer.
answer. Please don't say the answer. We'll
Okay, okay,
discuss further and we'll finalize the answer. Uh,
yeah, okay.
yeah, Kritika, you were saying something. Can you repeat, please?
Yes, you were asking a question like what is random
Yeah.
mutagenesis? Sir, it's like when we talk about the evolution, so
Hmm.
we said there are some errors or mutations which cause the mutation. So that is random mutagenesis, which takes time to
Hmm.
mutate one organism or species, and it's take a long time. It's not like we will just target a specific target and it will cause mutagenesis. No, it's not like that. It's like it's not decided where the mutation will occur.
Huh. Okay, so just a clarification. So, based on what Swati told and Kritika told, uh, random mutagenesis, of course, is not focused or concentrated on mutating a particular enzyme or protein, but rather you expose your organism of interest to a mutagen that can cause any kind of changes. And does anyone want to answer how is mutagenesis, how was random mutagenesis used in metabolic engineering previously? Does anyone want to answer on that aspect?
Uh, Production of penicillin.
I mean, like that is, you can go further. Explain how do you think random mutagenesis was used?
So it can generally use to explore the genetic diversity between the microorganisms which have a better or a efficient metabolic pathway.
So you can talk about it further. Would you want to discuss further about it?
So this is what I know.
Okay, no issues. So earlier, when molecular biology techniques were not discovered or not very well adapted to specific organisms of interest, what scientists would do is they would take an organism and expose it to mutagens, and that would give rise to mutations, which you don't see directly. But then you start screening all these different mutated organisms and check if any of these have higher yield or higher titers of your desired product. Then, based on that, you go back and do genetic sequencing to identify what possibly caused the mutation. So, that is random mutagenesis, and that is its relation to metabolic engineering. And uh, does anyone want to answer this question now that we've discussed what random mutagenesis and how it was used for metabolic engineering? Yeah, you can just switch on your mic and say the answer. Anybody?
So the answer will be B,
False.
false, because
Okay.
in metabolic engineering,
Huh.
we modify a specific target to get
Okay.
a specific product which we require.
Huh. Yeah. Do others agree to it?
Yes, sir.
Okay. Huh. So the answer is false. Random mutagenesis is not the focal theme of metabolic engineering. Yeah. The next question is recombinant bacterial cells can host genes from another organism which have A. Bacterial origin, B. Archaeal origin, C. Eukaryotic origin, D. All of the above. We can take a few minutes and then you can raise your hands and then start discussing what you think about the question. Swati, you can go first in a couple of minutes. Yeah, Swati, before you go to the answer, you can first say what you think about genes from different organisms of different origins. How do you think genes from bacterial origin vary from genes from eukaryotic origin? You can first share your thoughts on that, then we'll discuss what the right answer is.
So if we take the example of insulin production,
Hmm.
we are actually introducing the gene which produce human insulin into E. coli.
Right.
I think similarly, so using microorganisms or bacterial cells or E. coli cells, you can produce human uh, proteins if you introduce human genes
Ha.
into bacterial cells. Similarly, if you have to change the bacterial, like you know what is archaebacteria is, right? The archaebacteria are actually, I think it's a prehistoric form of bacteria which can survive in extreme conditions like excessive acidic condition, temperature, etc. So if
Hmm.
you have to bring such a condition to the bacterial cell, then you can take that particular gene responsible for uh, surviving in extreme conditions into this bacteria so that you can bring about that particular feature to this bacterial cell.
Hmm. And normal
I mean,
bacteria, you can. Okay. Uh, You're partially right. I would explain it further if does anyone else want to share their thoughts. Kritika? Yeah, you can go ahead, Kritika.
Sir, can you please repeat the question again?
Yeah, so the question is recombinant bacterial cells can host genes from another organism which have, and you have three different options: bacterial origin, archaeal origin, and eukaryotic origin. What I wanted to focus on here is, do you guys think if the genes from bacterial origin or archaeal origin or eukaryotic origin will have any differences in them? At the gene level, is there any difference between an eukaryotic gene, an archaeal gene, and a bacterial gene?
Uh, when we talk about the bacterial origin, so
Hmm,
these are prokaryotes, bacteria and archaea. So
right.
when we talk about the bacteria, they lack like introns and exons, which we have in our eukaryotic origin. And in archaeal, I think these have introns just like eukaryotic origin. But when we talk about the gene, I think they have universal genetic code in them. So I think these three options can be considered to be uh, which recombinant bacterial cells can take. So I think option D can be the answer.
Okay. So, Ayush and Surendra, do you want to share your thoughts on this? Like, how do you think genes from bacterial origin, archaeal origin, and eukaryotic origin vary?
Bacterial and eukaryotic genes vary. Differences.
Yeah, the differences between genes that you observe in a bacteria and the genes that you observe in a eukaryote.
Main is the introns are present in the eukaryotic genes,
Hmm,
but they are absent in the prokaryotic genes.
right.
And eukaryotic genes having the poly A tail, and it is not present
Hmm.
in case of the bacterial
Just a correction, polyataid, you get
genes.
that in the mRNA. You don't have
Uh-huh.
that in the
mRNA, yeah,
genes.
yeah, yeah, yeah. Yeah.
So that
Uh-huh.
that is not right. Huh.
Okay, okay.
So what I just wanted to mention, Ayush, do you want to say, you want to contribute to the discussion or should,
So we have already talked much detail about it, so I think we can continue.
huh. Okay, so the, as you guys said, there are differences between bacterial and eukaryotic genes. Yeah, it's true. Prokaryotic genes are polycystronic, true, as Swati mentioned in the chat box. So what we need to consider is that independent of the origin of the gene, if the uh, like in case of eukaryotes, you have an intron, an exon, and then an intron and an exon. So internet issue. போட்டிருக்கேன் hotspot. Yeah, sorry. Because of my internet issue, I'm switching off my camera. I hope you can hear me.
Yes, sir, it's okay.
Yeah.
Yes,
Is,
sir.
is okay. My screen. Yeah, is my screen visible?
No,
No,
sir.
sir.
The screen is invisible. One moment. I've shared it. Yeah. I hope my screen is now visible.
Yes, sir, yes.
Yes, sir.
Yeah. Yeah. So as we were discussing, genes from bacterial origin do not have introns, they only have the coding region. But genes from eukaryotic origin have both intron and exon. And genes from archaeal origin, like one of you mentioned, I think it was Swati, that they grow in different conditions, which are extreme conditions like high temperature or high salt conditions, or could be acidophiles. So, in order to counter that, their genome will have high GC count. That is a particular characteristic of genes from archaeal origin. With respect to hosting genes from other origins in bacterial cells, what one has to ensure is that these genes are protein producing or enzyme producing genes. So the proteins have to be synthesized. So if proteins have to be synthesized, because bacterial cells do not have the machinery to split or identify introns and exons, when genes from eukaryotic origin are put into bacterial cells, all the protein coding genes like exon 1 and exon 2 and exon 3, all of these have to be stitched together and these have to be introduced. So that is something that one has to take into consideration. So, like you all answered, the answer is all of the above, but there are certain limitations or certain considerations that have to be taken care of, like the one that I mentioned. The next question is, uh, yeah, sorry, was the answer clear?
Yes, sir, it was clear.
Okay.
Yes,
The
sir.
next, thanks. Next question is, E. coli has been widely used as a microbial cell factory because of the following factors. 1 minute. Uh, and the options are high growth rate and high cell density, wide availability of gene based models and genome engineering tools, and biopharmaceuticals are largely derived from E. coli cells. So we can wait for a couple of minutes and think about it. Someone new has joined. Uh, Priya Maria. I'm Shankar. I will be the TA for this course. We'll be meeting on Mondays from 6 to 8 and discuss queries if you have any. And I'm a PhD student from ISC Bangalore. You can introduce yourself, Priya Maria.
Hi sir, myself Priya. I'm studying MSc Biotechnology at the University of Madras.
Okay. So we have covered most of the questions earlier, Priya, but we'll once we finish all the questions, we'll again go through them for clarity, or you can also take a look at the recording. Yeah, Kritika, you wanted to uh, share your thought on this question. Why do you think E. coli has been widely used as microbial cell factory? You can unmute and you can start sharing what you think. You can talk about one point specifically. We'll give chance to others as well.
Okay, sir. So I'll explain first point. So
Huh.
we have performed so many experiments in our lab as well. So I have
Okay.
seen that E. coli can grow very fast. They have replication time of 20 minutes, I guess.
Huh.
Yes, 20 minutes. And they have high cell density. So option one is correct, and rest other can explain, sir.
Yeah. Uh, yeah. Priya, would you want to share what you think? Do you think any of the options? Yeah. Priya, are you there? Okay. Then, Surender, would you want to share your view on any of the other two options?
Mm. As earlier discussed, the growth rate of the E. coli is high. Uh, it means their expression inside them, expression of genes is also high, and the product will be produced more there. And moreover, genome of the E. coli is widely studied. Approximately 60-70% of the genome of the E. coli is already studied, and variety of the molecular biology tools are available to study the E. coli, and which are not available for the other organisms.
Okay.
And mostly, as discussed in the lectures also, mainly mammalian cells are used for the expression purpose, na? But E. coli is 23% of the pharmaceuticals are produced in the E. coli strains by industries. Thank you. Thank you.
So just a correction, like you mentioned, E. coli has higher
Yeah,
growth
yeah.
rate, but that does not mean that it has higher expression rates. So expression is independent of the growth.
Okay.
So what matters in metabolic
Okay.
engineering or
Okay.
in microbial cell factory is that if you have higher growth rate, you'll have higher biomass. You'll have many number of cells, so
Yeah, yeah.
that would equate to higher product formation and not the expression. Expression you would alter using molecular biology techniques like we previously discussed. And the answer here is all of the above, like you guys mentioned.
Sir, can I add one more point?
Yeah, sure.
Sir, like you said, that expression is not dependent on growth. If we want to see the expression of a particular gene in E. coli, that means the enzyme which is encoding for that gene
Yeah.
should be present there. Then only it can express that gene. If there is no such thing is there in the E. coli, then it won't be able to express that particular gene which we want to study.
So that is where the heterologous expression part comes. So you don't have a particular enzyme that is naturally there in E. coli, but you can take that enzyme, take the coding genes, put it inside a plasmid, and introduce this plasmid in E. coli. Now you have an E. coli which has a heterologous gene, and then you can express this.
Yes,
But
sir. That's the
but
point.
I didn't understand why you had mentioned that here.
No, sir, this was my query when I have gone through the lectures. So this was my query. That's why I've raised this point here.
Okay, okay. So that is called heterologous expression, like we, independent of the origin and independent of what gene is it, as long as you have the coding region, you can put it inside a plasmid, put it in front of a promoter, and in the form of an operon, have an antibiotic resistance marker so that you can select gene. Yeah, sorry.
One point I can add here.
Yeah, yeah, sure.
As you said, growth and gene expression, they are not related to each other.
No, I didn't mention that growth and gene expression are not related to each other. What I mentioned is
Huh.
that higher growth rate does not necessarily mean higher expression.
Yeah, yeah. But for the growth, na, proteins are required.
Yes,
For cell
proteins are
division, proteins are
required.
required.
Yeah,
And these proteins are coming from the expressions, gene expression.
yeah. What I meant is higher expression of a particular gene that we are interested in does not
Mm.
automatically come with higher growth rate.
Okay, okay.
Let's say you have a gene. Okay, so in
Mm-hmm.
metabolic engineering, like we were discussing, you take a gene, then you put that inside a plasmid, and all of these have something called as an inducer. In most cases this is a sugar analog, and we use something called IPTG. So this binds to the operator region and it
Inducer.
helps
Yeah, yeah,
in
yeah. Got it.
overexpression. So independent of the
Right.
growth rate, you can express more of this.
Increase.
Yeah.
Yeah, yeah. Got it. Thank you. Thank you.
Huh. So the
Sir,
answer
can you
here,
explain one thing,
yeah, sure.
why high cell density is an important factor in this?
Okay. So when you consider, okay, when they talk about microbial cell factory, they don't talk about experiments at the lab scale. Rather, they talk about experiments at the industrial scale. And when you do at the industry, you have large fermenters that are about 50,000 liters in capacity. So you have a system where you have these cells growing in a fermenter. When you do such a large growth at such a large scale, you would want a large number of cells. That is the reason you start with a 50,000 liter capacity fermenter. And when you have such a large fermenter with many number of cells, the amount of biomass, which is the cell alone without the media, that will be a lot higher. And inside these cells is your product of interest. More the number of cells, assuming equal expression in all cells, more the number of cells would give you more product than lesser number of cells. That is why high cell density is important. Was that clear or okay?
Yes, sir.
Okay, thanks. And the last question is just a generic question. Just the first licensed drug produced using recombinant DNA technology. Uh, we'll just wait for 1 more minute and then one of you guys can discuss and then we go. Yeah. Swati, you can start by explaining one point and then we can go ahead with someone else.
Sir, I think the answer is option
B.
B.
Okay. It's straightforward. Okay. So these are set of sample questions that I have prepared for this assignment. Surinder and Ayush were there for the entire set of questions. Kritika and Swati, you had missed a few questions. So I thought we can just go through them before you leave. So uh, we'll start with Swati. So, uh, um, I think you joined us when we were talking about the molecular biology techniques, if I'm not wrong. We have
Yes,
about four
actually,
questions to go.
I
Yeah, sorry.
reached home at 5:30 p.m. from college. So that's what.
No, no issues, no issues. We'll just go through these questions, we'll discuss them,
Yeah,
and then so that it will be helpful for you. And Kritika, you had also missed a few questions. So I'll start from the beginning so that it's beneficial for all of you. Uh, so the first question was, what does not hold true for computer-aided synthesis of biochemical pathways? Be nice. The options are: it considers the chemical structure of the substrates and products, it has a non generalized algorithm, it does not consider the enzyme rules, and de novo design of enzymatic pathways. So, the question that's being asked is for uh, what does not hold true. You can just take a couple of minutes to think and then you can answer, Kritika. Yeah, Kritika, you can answer. You can share your thought and we can try discussing the answer.
So basically, option one is correct because this be nice consider the chemical structure of the substrates and products.
Okay.
Option B is incorrect because it has generalized algorithm, not the non-generalized. And C is also incorrect because it does consider the enzyme rules, but it's written here that it does not consider the enzyme rules. And D is de novo design of enzymatic pathways. Option D is correct. So I will choose option E, that is B and C does not hold true for B nice.
Okay. Swati?
actually, sir, I don't know the answer for this question. That's what
Oh,
I didn't raise my hand.
okay. Okay. So, uh, yeah, like Kritika had mentioned, um, Be Nice is actually a server, web server. If you type benice.ch, you will be able to reach that server. So, this server is used to analyze pathways, and the server is made out of an algorithm that applies to all pathways and not a specific pathway. So it is a generalized algorithm. And because it works with pathways, it has to deal with substrates and products. So the chemical structures are incorporated, and what kind of enzymes can act on what kind of substrates and products are also incorporated. That is the enzymatic rules. And with this, you can also try and see if new pathway that did not exist in a particular organism can come up given a particular substrate or an enzyme. That is called design of de novo enzymatic pathways. So, like Kritika mentioned, all these out of these four options, option B and option C are false. So the right answer is option E, B, and C. Uh, Swati, was it clear, or should I try explaining it?
Yes, sir, kind of. Like, I think this is the part which comes under lecture 5, right?
Yeah, so this is a part which comes under lecture 5, minute 14. You can even take a look at the video.
Okay, sir, I actually haven't. I have seen till lecture 4 only. That's what I got.
Okay, okay, no issues. You can take a look at it and you can also post a query if you have in the discussion forum.
Yes, sir. Yes, sir. Thank you for explaining.
So the next question is seed metabolites refers to option A externally acquired molecules, option B internally synthesized molecules, option C the first metabolite of the rate limiting step, and option D none of the above. Yeah, so we can just wait for a couple of minutes and then think about the question. Swati can go first after a couple of minutes. Yeah, Swati, you can try sharing what you think.
Sir, I think the answer for the this question is option A, uh,
Mm-hmm.
because seed metabolites are something which you provide externally. It is
Mm.
not produced within the cell.
Okay.
Uh, yeah. So I have studied this, like, in chemistry also, in coagulation. When we learn coagulation concept, I have studied that when we add something to the solution, the thing which has to be precipitated will get start coagulating over this added substance. So it is that process called seeding also. So
Ah,
similarly, I have connected this one.
okay. Mm. Uh, so seed metabolites are like you guys mentioned, uh, are not naturally available or produced within the cell, and these are needed for the pathway to start. So usually these are the first set of metabolites that enter the pathway from the media or environment, and the pathway starts. And further, because of the action of enzymes, these get converted into different products. The answer is externally acquired metabolites. Uh, Hi Srihari, I am Shankar. I am the TA for this course. We'll be meeting on Mondays from 6 to 8. Uh, I'm a PhD student from ISC Bangalore. You can introduce yourself. Yeah, Srihari, are you there? Okay, if not, let's go to the other question. The third question is what is heterologous expression of gene? The options are expression of gene or part of gene in host organisms that naturally have the gene or gene fragment, expression of gene or part of gene in host organisms that naturally do not have the gene or gene fragment, both A and B, and option D, none of the above. So, yeah, Swati, you can try sharing what you think.
Sir, I think I was there for this question.
Oh, you were there.
Shrutika
Okay.
was there.
Okay, Shrutika has left. Okay. If that is the case, um, these set of questions are over. Other than this, you should have received an email with assignment 0. Did you guys receive an email?
Yes, sir. I think we have to. Yeah, we will get a PDF of the solutions, right?
Yeah, so in case, since we have finished discussing, sorry, is Kritika there? Okay. Huh, sorry, I thought you had left. Yeah, Kritika, do you want to answer this question? What is heterologous expression of G?
Yes, sir. It's option B, which is not naturally present in the host, sir.
Okay. Yeah, that is right. That is right. And were you there for this question, Kritika?
Sorry, sir.
Did you join the meeting for this question? Which of the following are not involved in the production of microbial?
No, sir.
So then you can maybe think and try to reason out this what is the right answer for this question. Take a couple of minutes and then try to answer. Kritika, do you want to answer?
Sir, I'm not sure.
Okay, you can tell us what you're
I
not sure. We
think
can discuss.
it should be option D, sir.
So you think bioinformatic and mathematical modeling and the flux redirected approaches are not involved in microbial cell factories, is it?
Yes, sir.
Okay. So microbial cell factories are these microbes where you produce your products of interest, right?
Yes, sir.
So to be able to produce your product of interest, you need to remove competing pathways. So let's say your pathway consists of 4 5 steps, 4 5 substrates, I mean 4 5 intermediates. And these intermediates also can branch out to different products. Um, in those cases, to get your product of interest, you need to decrease the number of branching so that more amount of your product is being formed. That approach where you determine how to reduce the branching of your pathway is called flux redirection. And this is important in microbial cell factories because this is one way you can maximize the amount of product being formed. And other than this, bioinformatic and mathematical modeling, where qualitative analysis of pathways is done. So here, by bioinformatic, what is meant is that you compare pathways from different organisms to produce the same product. This way, you'll get an idea what is the minimal number of steps, minimal number of enzymes that is required, and mathematical modeling here means how to quantify the rate at which the enzymes work. So let's say one enzyme works fast, one enzymes work slow from different organisms. Through mathematical modeling, you'll be able to say with this much substrate, this much product will be produced by this enzyme. So this is faster, so I can use this. So that way, both A and C are important, and of course B is important. So the right answer for this question is none of the above. All the three options shown here are important and is involved for production of microbial cell factories. Was that clear, Ritika?
Yes, sir.
Okay. So and I, Kritika, were you there for this question? The molecular biology techniques part.
Yes, sir. I've answered this question.
So with that, we cover the set of questions that were designed to help you solve week one's assignment. Other than this, I have the other PDF, which is assignment zero solution. So you would have received an email for this. Since we have about 15 minutes left, I thought we can also go through these questions, might be beneficial for you. So we can quickly go through them. Uh, I hope you are able to see what I'm sharing.
Yes, sir.
Yeah. So uh, you can take turns. Whoever want to answer can try explaining, or if you have any doubts, that is also welcome. So the first question is, which one of the following terms best describes the cellular process of breaking down large complex molecules into smaller ones with the release of energy? So since the answers are available here, I would want you guys to rather discuss or justify why particular answer is right rather than just stating an answer. Like for example, in question one, there are the four options of catabolism, biodegradation, anabolism, and dehydration. So if one want to answer, they can try answering why it is called catabolism and not biodegradation. Does anyone want to answer this? Okay. So first, yeah, okay, since Kritika is raised, who was raised? Yeah, Kritika is raised. Yeah, you can go ahead, Kritika. Next, we'll have Swati.
First question, it's asked which term describes the cellular process of breaking down the large or complex molecules into
Um,
smaller ones.
yeah.
So, first is catabolism, which is the specific term for breaking down the large molecules into the smaller ones. While we talk about anabolism, that means small molecules they attach together to form a large molecule.
Huh.
Dehydration means removal of water molecules. The
Huh.
option is first A option.
But what about biodegradation?
So degradation means, like I can take one example. We have translated a gene into a protein.
Okay.
So after post-translation modifications with are there, for example, ubiquitination.
Huh.
So we are not using any kind of chemical there, but it's a biological method how we are degrading that protein which is not useful to us. So that is biodegradation.
Okay, just a clarification. So the question here asks you uh, the process of breaking down complex molecules into smaller ones with the release of energy. So that should also be taken into account. Yeah, Surinder, you can um, share us what you think.
In biodegradation, energy is not released
Huh.
and it is used for the growth of the but bacteria used this for the growth purpose.
Right, that is true. That is something that I wanted to explain. So, Kritika, just a correction, biodegradation is also a process where uh, large molecules can be broken down into smaller ones. But what happens here is instead of just using the enzymes, you use microbes in certain cases, and for microbes to grow, like how Surender had mentioned, they need energy. So biodegradation, despite breaking down the large complex molecules into smaller ones, it takes in energy but does not release energy. That is why the right answer here is catabolism.
So I have a
Yes.
doubt here.
Yeah, sure.
Biodegradation, like, can we consider this one as the example, right? Like the breakdown of organic molecules or waste materials by microorganisms, can it be considered as a biodegradation?
Yeah, that is biodegradation. That is
Yeah.
right.
Okay. Okay. Okay.
Huh. Yeah, Kritika.
Yes, sir, I got it.
Okay. Huh. Then the second question is, which of the following points is applicable for bacterial inner membrane? So, Swati. Yeah, you can go ahead, Swati.
So bacterial inner membrane, I think bacteria also have a cell wall, peptidoglycan cell wall. So here the question is about bacterial inner membrane, which is also a phospholipid bilayer. And it has the same characteristic features as that of eukaryotic cell membranes. It is semi-permeable, it is bilayer, and this I think the cell membrane of bacteria also possess proteins, transmembrane proteins like that of eukaryotic cell membrane. So I think all of the above will be the answer. So
Right, that's right.
option B.
Yeah. The third question is, enzymes catalyze specific reactions by option A, lowering the activation energy, option B, affecting the reaction rate, option C, changing the reaction equilibrium, option D, both A and B, option E, both B and C. So, does anyone want to answer? You can raise your hand. Ayush?
Question D.
Option D, both A and B. Okay,
Yeah.
that's. Uh-huh. Uh, Swati and Kritika, what do you think?
Sir, it's D.
Ayush thinks it's D.
So
Swati?
because low as enzyme catalyze the reaction, it lowers the activation energy, so it able to reach the threshold, uh, and threshold level. So it uh, directly affect the reaction rate, which is uh, which is fasting the rate of reaction.
Can you repeat again, Ayush?
So, as the enzyme catalyze the reaction, it
Huh.
lowers the activation energy so that it able to reach the threshold level, which with the minimum use of energy, and which generally affects the rate of reaction.
Okay. Swati and Kritika, do you think it's option D?
Yes, sir, I completely agree with Ayush, sir.
Okay.
Yes, sir, same here.
Okay. You can also go check if enzymes directly affect the reaction rate. They definitely affect the activation energy. They lower it, but you can also check if it directly affects the reaction rate. You can go read about that. And for question 4, which of the following points are most appropriate for fermentation? Does anyone want to maybe answer this? Why it is? Yeah, go ahead. One. Yeah, Kritika first.
Sir, only option A is correct because when we talk about option B,
Huh.
so if there is presence of oxygen,
Huh.
then fermentation somehow will not occur because as we generally see the bread making, it forms by fermentation. So CO2 evolve, which gives its formation. So, like in lack of O2 or in limited amount of O2 can be accepted in that way. And C is it allows complete oxidation of carbon substrate. Then, sir, it basically and does not support the fermentation, because in fermentation, as we know, it forms ethanol or other products. When we talk about the complete oxidation of carbon substrate, then it will form CO2 and H2O, which is a uh, basic, I'll say, respiration process.
Mm-hmm.
Yes, sir. So it will be option A, in which the endogenous organic compound act as a electron acceptor.
Okay. If others agree to this, we can go to the next question.
Yes, sir.
Yeah. Okay. Huh. So the next question is, which of the following statements is or are true about enzyme catalyzed reactions? Option A is the reaction is faster than the same reaction in the absence of enzyme. Option B is the free energy change of the reaction is opposite from the reaction in the absence of the enzyme. Option C is the reaction always goes in direction towards chemical equilibrium. Option D is A and B only. So, yeah, since Swati is hands raised. Ha, Swati, you can answer.
So about enzyme catalyzed reactions, we know that in presence of an enzyme, the reaction rate would be faster. The reaction will occur faster than in the absence of enzyme.
Right.
And so option A is the right answer. And as for the second option, free energy change in the reaction is opposite to the reaction in the absence of the enzyme. It cannot be the case, sir, because for a spontaneous reaction, free energy change is always negative, right?
Right.
So in the absence of the enzyme, the reaction takes place. So it cannot be the opposite. Opposite means it can suppose the Gibbs free energy for a reaction is minus 10. Suppose
Right.
according to the statement, in the absence of the enzyme, the Gibbs free energy will be plus 10. But that will not be the case, because in the absence of enzymes, the small amount of product can be produced, but it may take time. Doesn't mean that it won't take place. So Gibbs free energy can't be positive in that case. That is my take to that second option. And third option, the reaction always goes in the direction toward chemical equilibrium. I am having some, I need some clarification there, but I know that
Okay.
first two obviously.
Okay, so what we mean by chemical equilibrium is when the amount of product, the concentration of the products and the reactants are the same. That is a state of equilibrium. And ideally, your enzyme catalyzed reactions would want to convert most of the substrate to product. Thereby, you will not have a balance of the substrate and the product, but you will have more of the products and very little of the substrate. So it is actually not, it does not go in the direction towards chemical equilibrium. It will achieve equilibrium and it will go beyond equilibrium towards product formation. So that is why option C is wrong. I
Okay,
think
sir.
there
Okay,
was
thank
lag.
you.
Ah, yeah. Then,
So,
yeah, yeah, Surinder. Or
you mean that all of the substrate, 100% of substrate can be converted to the product?
given enough enzyme, 100% of substrate can be converted to product.
Okay. If enzyme
Huh.
is enough.
Yeah, if so, in all of these cases, the rate limiting factor is your enzyme. In any kind of enzymatic reaction, you will have very little of enzyme, a lot of substrate. So,
Yeah, yeah, yeah.
the number of times an enzyme can interact with the substrate determines how much product is being formed. Let's say if we have 1 unit of enzyme and 1 unit of substrate, then definitely it will be converted to the product and you will only have the enzyme and the product left. So that is what I wanted to mention. The sixth question is active transport of nutrients across biological membrane is characterized to be. So again, if anyone wants to answer, you can answer. We'll discuss it. We're talking about a particular transport called as active transport. There are two kinds. One is active, one is passive. One requires energy, one doesn't require energy. One requires energy because it has to transport against a concentration gradient. So that is what is mentioned here. So the options include requires energy and occurs against the concentration gradient. Option F says that both these are right, so that is the right answer. And option 7 is that which of the following is an electron carrier in metabolism? ATP, ADP, peptidoglycan, and NADH. Does anyone want to explain what an electron carrier is? Yeah, sorry.
Which can be reduced,
Huh. Which can it be
reduced.
oxidized?
Yeah, can be oxidized and reduced both.
Okay, so basically electron carriers are those intermediates that can take up an electron or give up an, and that way they will help in maintaining the charge balance within the system. So in this case, ATP and ADP are energy currencies. Peptidoglycan is something that's a part of the cell wall. NADH is the right answer. It acts as an electron carrier. You might have studied this in your uh, different cycles.
Can I
Yeah.
ask one question here?
Yeah, sure, sure.
In sixth question,
Sixth question, yeah.
why not A is not there? Active transport carrier is required
Carrier is required.
for active transport.
Sir, can I
Uh,
answer this question?
yeah, you can.
So basically, these carriers are required in the facilitated diffusion. When we talk about the ATP, so there is a utilization of ATP when we talk about the active transport. So if you have studied about the primary and secondary active transport, where there is transportation of sodium, glucose, or sodium potassium pump, these are the example of active transport.
But some carrier is required for making of the molecule to be transported inside or outside the cell.
So,
Basically,
yeah, sorry, you can go ahead, Kritika.
for active transport, you don't require these carrier. So basically, there is co-transportation. If you have, like, for example, sodium, it can move from a higher concentration, sorry, a lower concentration to the higher concentration, and it utilizes the ATP. And with this, glucose will move as well in the same direction. So it doesn't require this,
But
but
they move,
uh,
but they move through a transporter, no? Some transporter, and
There
that
are certain
transporter is a carrier.
pores.
That is a
Uh,
carrier.
yeah, sir. There are certain pores in the membrane across which these molecules will pass through. I think so. So carrier molecules are not required. So to open these pores, we may need certain molecules like ligands, so-called calcium ions or magnesium ions or whatever may be to open the pores so that these molecules can go cross the membrane
Just,
utilizing the ATP.
just a correction. So, if you have pores in the membrane and if ions can passively diffuse, then you don't need energy. All you need is a concentration gradient. They'll travel from higher concentration to lower concentration so that they reach the same concentration. That is called as the chemical equilibrium, which we previously discussed. So that is not factually right, Swati. So what um, is the discussion here is whether active transport always needs carriers. And as far as I know, I think active transport the word active here describes the necessity for energy and the presence of the absence of a concentration gradient. So basically, you are transporting an ion from one region to another region, from a region of lower concentration to higher concentration. Naturally, this wouldn't occur because higher concentration has enough. So you don't need to provide more of the ion. But in order to do that, you need to spend energy. That is why it's called active. I am not sure about why carrier mediated is not mentioned, Surender, but I will get back to you. I'll check and I'll get back to you.
Okay.
Huh.
Okay, sir.
Yeah. And the last set of questions, which of the following is not a polymer? Glucose, starch, cellulose, and chitin. We all know that glucose is the basic sugar. So, in order for it to, this is what leads to polymers like cellulose, starch, and others. Which of the following options best describe the term metabolism? So, all catabolic reactions, all anabolic reactions, both catabolic and anabolic reactions, and finally, all biochemical reactions. Does anyone want to answer why it's option D and not option C?
It may be transformation,
Huh.
conversion of glucose to fructose. It is
Right,
a biochemical reaction.
right. Yeah,
Neither it
that
is a
is right.
catabolism nor it is anabolism.
Right. You're right, Surinder. So, apart from breaking down or making up of complex or simple molecules, you can also have interconversion between them, like Surinder had mentioned. And so, all biochemical reactions within a cell constitute the metabolism. And yeah, identify the most appropriate energy carrier or currency of the cell, and we know that ATP is the energy carrier. So, with this, we'll end today's discussion. Thank you for attending. Next, we'll meet on next Monday, same time at 6 to 8. So, you can solve your assignments, and we'll be discussing some sample questions that will help you solve assignment 2. Thank you for attending.
Thank you.
Thank you, sir.
Thank you.